lecture with regard to the topic of epigenetics. I am very involved with sea urchins and ocean acidification. So, towards that topic, I am interested in actually there is a chance to accomplish a proposal that is volcanic vents as a natural laboratories to understand the urchin adaptation to ocean acidification. I want to make clear that I am an aquaculture. I am not a scientist of lab, so this is a whole new topic for me, but I am really enjoying it. So, which one is the objective of this endeavor, of this proposal? Is to identify the SNPs or SNAPs related to ocean acidification adaptation of targeted species. In this case, is sea urchins. But what is the special key here? We're talking about sea urchins in Galapagos. What is happening in Galapagos? In Galapagos, we have volcanic seeps. What are volcanic seeps? Volcanic seeps are these structures that naturally bubble continuously CO2. So what is the outcome of this? This is a natural laboratory. So all the species that live in these areas are naturally adapted to acidity. So this is great because it's a natural laboratory in the backyard. Okay? So what are the expected results? An update and a reliable library for sequencing from the target species, that in this case is, as I said, sea urchins, in order to evaluate which are the epigenetic factors that make these species adaptable, resistant to these conditions, okay? So, in order to structure, from the aspect of epigenetic, in order to structure this project, I searched for some investigation and I found a very interesting paper from Dr. Samuel Utik in 2019, it's published, and he actually picked up information for individuals endemic of these seeps, right? And he made a comparison between another situations and other conditions. So he established this experimental design. We have these control individuals and these vent individuals. We can see there the nomenclature, the C and the V, right? So he extracts these individuals and use them to spawn, no? And he has the opportunity, his team and everything, has the opportunity to evaluate what is the degree of resistance of the larvae and the Plurius larvae. Why is it important to evaluate the early stages of the organism? Because it's most vulnerable, okay? It's the most vulnerable in such conditions. So, if I can recall the techniques that he implemented, is that the main goal for him was to extract the library, the genome, because there is information that is going to give us answer in order of the behavior. So, and here we are going to see a little bit of the recipe, the mechanics of the situation. The first thing that he does is the destruction of the sequencing. He uses RNA layer, that is a solution, in order to extract information that we want to evaluate, filtrated and depurated, and then the Illumina sequencing is the key in order to obtain the library. There is information that we need. After that, we create a de novo transcriptome. Why is de novo transcriptome? Because there is no previous record of such information. So this is what I think, what I might need to do in order to replicate this kind of work, but with Epigenetic galapagensis. Okay, but this gets more exciting. Why? Because what are the future perspectives for agriculture? All the organisms are facing what? Ocean acidification or illness, parasites. So with epigenetic, we have this choice option to discover what are the tools, what are the alternatives to overcome such circumstances. Okay? So in order to potential implementation of such tools, such alternatives, we can as well implement better practices toward agriculture. Okay? Because epigenetics provide the potential of custom design traits as behaviors, and then adaptability on performance based on ecological systems. As we can see here, we can see a system, a multi-trophic system, which every one of the cultures accomplish a function. There is no competition. There is improvement. Okay. So, what is the potential implementation of this? Well, the campaign in Galapagos, that is a potential choice, is a potential alternative to make a comparison study between species in Ecuador, species in Chile, in Spain, and in Argentina. This is a group work. This is everyone is involved. So, what are the conclusive remarks here? To improve agriculture production system, epigenetics share as a window of reliability towards expected performance and the option to look for answers. Therefore, more incentives should be promoted and favored to sustain development and improvement of such analysis tools. Those are my references, and if there's any question.
Very nice, very nice. So you're going to use the natural populations that are exposed to these conditions and identify what makes them well adapted.
Yes.
And so then can you take it through, once you get that information, what's the next step? Once you make all these discoveries, how do you implement the discoveries? Yeah.
Well, I think that the process is to extract adults, then spawn them in controlled conditions, then work with the juveniles, with the larvae stages, and establish a factorial design to compare multi-stressors. And the outcome of the response, we get the genetic information in order to get library and test again the results in the realm of the genetic. Yeah, that's what I think.
No, that's good. Nice job, very good.